Hello, my name is Roderick L. Roll, and today I will be talking to you about antibodies, more specifically, how we utilize antibodies in research and in medicine. How can we use these antibodies? In the slide, we're looking at a 96 well plate. This is nothing more than 96 very tiny petri dishes. In the bottom of each well, here is well number A. In the bottom of that well, you're going to have a flat portion like a petri dish. And inside of those wells, we're going to label it, we're going to tag it, we're going to adhere to the bottom either an antibody or an antigen. Enzyme-linked immunosorbent assay is referred to as an ELISA test. This is going to utilize antibodies and enzymes. Collectively, the enzyme is going to be attached to the antibody and a colorimetric reaction is going to occur via the enzymes. So if you look at this portion here, which is the substrate, it is going to be converted into a product. What will catalyze that reaction is the enzyme, and the enzyme is linked to an antibody. This process of actively labeling our antigen is enzyme-linked immunosorbent assay, which is called an ELISA test. The first step is to immobilize the antigen. You're going to immobilize the antigen onto a plate. Remember, we're talking about a petri dish that is just miniaturized. So the 96 well plate is just 96 very, very tiny petri dishes. So the first step is to immobilize the antigen. I can immobilize it indirectly or directly. Indirectly, we're going to first use an antibody that will be bound to the petri dish. This is our capture antibody. That capture antibody is going to capture the antigen. Once it captures the antigen, then the second antibody will bind. This creates a sandwich. So, indirectly, the antigen is immobilized. Now let's look at the direct method. Directly, the wall of the plate will have this antigen. Here is the antigen linked directly to the bottom of the petri dish, bottom of the 96 well plate. Therefore, the antibody can bind to the antigen. So those are two ways to immobilize the antigen. That is step one. Step two is to use a detection antibody. Directly conjugated to an enzyme such as horseradish peroxidase. In this first image to the left, we're looking at horseradish peroxidase. That is an enzyme complex that is going to create some type of color. And attached to that horseradish peroxidase is our detection antibody. So we're looking at detecting the actual antigen. Here is our antigen. It could be a virus. It could be a part of a virus. It can be a part of a bacterial cell wall. So the detection antibody will bind directly to that, and it will alert the researcher that something is there. How will it alert it? It will alert through an enzymatic reaction. This enzyme is conjugated, it's stuck to the antibody. The second method of detection antibody is indirectly. Indirectly provides a binding site for a labeled secondary antibody. So let's look at the second picture. Here's our detection antibody. It is actually bound to the petri dish, not like the other one that was bound to the actual antigen. Now we have a secondary antibody that will bind to the primary antibody. And the secondary antibody has the conjugated enzyme. And that enzyme is indirectly going to create this color that we can detect. So those are two separate methods for using detection antibodies. ELISA can be subdivided into four separate types of ELISA. We have the direct ELISA, we have the indirect ELISA, we have sandwich ELISA, and competitive ELISA. We will first discuss the sandwich ELISA. This method is going to utilize an antibody and adhere it to the bottom of a petri dish, or in our case, we're using the 96 well plate. Then the antigen of interest will bind to that antibody. The antigen can be a fragment of the coronavirus. It can be a fragment of the Ebola virus. It can be a fragment of a bacteria like Staphylococcus. So we have the antibody bound, and we have our antigen that hybridizes to that antibody. The next step is to take a secondary antibody and put it in the presence of the antigen. Now you have created this sandwich. This method is more effective in detecting low concentrations of antigen. A secondary conjugated antibody will have the enzyme linked to it, which will give us some type of colorimetric reaction. This can be detected by the researcher. These are some advantages to the sandwiched ELISA. High sensitivity. It's 2 to 5 times more sensitive than the direct or the indirect ELISA test. High specificity. 2 antibodies are involved in capturing. You have the primary and the secondary antibody that forms the sandwich. So it's more specific. And then flexibility. We have both direct and indirect detection can be used with the sandwiched method. The next method is indirect ELISA method. The antigen is coated to the plate. Down here, you see the antigen, the viral fragment or the bacterial wall fragment of a bacteria like staph will be coated onto the bottom of the plate. A primary antibody will be used to bind to the target. Here is our primary antibody binding to that antigen. Then a conjugated secondary antibody will bind to the primary. So here we have a antibody bound to another antibody, which is bound to our antigen. These are the advantages of that detection method. It's highly sensitive, economical. We have fewer labeled antibodies will be needed. It has a greater flexibility. Different primary antibodies can be used with a single labeled secondary antibody. The third method is the direct ELISA method. The plate is now going to be coated with an antigen. The patient's serum containing the antibody will be introduced to the 96 wells or to the petri dish. The patient, if they have the antibody, if they were previously exposed to this virus, then they will have the antibodies against the virus. The virus is attached to the bottom of the petri dish. The serum from the patient now is going to have antibodies that will hybridize to the antigen. Therefore, a colorimetric reaction will occur, and we will be able to tell if this patient has been exposed to the original antigen. So if the patient was previously exposed, they will produce the antibodies. An immune response will occur. We capture those antibodies, and they will bind to our antigen. The patient's antibody must contain a conjugated enzyme, like in this image. If it has the conjugated enzyme, then the colorimetric or the color detection will occur. This slide shows the advantages of using that method. It's faster than other ELISA tests. The technique has fewer steps. It's less prone to error. The final one we'll look at is competitive ELISA. First, coding must take place. Coding, we have control antigen is absorbed onto the plate. The next step, we need to remove liquid and wash the plate. The next step will be to add blocking buffer. A buffer containing unrelated protein is used to block free sites in the well. So this picture here has a blocking buffer. Then we're going to remove the liquid and wash the plate again. Prepare a sample of unknown antigen and detection antibody mixture. The next step is utilizing the sample. Add test sample mix to the well. We have an antibody that's already bound to an antigen. That was in our sample from the patient's blood. The next step, detection antibody. Add enzyme conjugated secondary detection antibody. So now that the antibody that was in the patient hybridizes right here to our antigen, we then add our secondary that is labeled. It will then bind to the original primary antibody. Then we have readout. Substrate is catalyzed by enzyme to generate a colored readout. So this is our competitive ELISA. The competition occurs here. These are the advantages for that competitive ELISA test. Let's recap. The enzyme is linked directly to the antigen. In this picture, here's our antigen. It is bound directly to the petri dish. Our antibody is the primary antibody. It has attached to it horseradish peroxidase. You can also use AP or GAL. Either one will take a substrate and convert it into a product that we can visually see. So this is directly linked enzyme assay. This is indirectly enzyme linked assay. So the enzyme here, horseradish peroxidase, is indirectly linked. Here is our antigen, and we have a primary antibody bound to the antigen. Our secondary antibody contains the enzyme horseradish peroxidase. So this is indirectly linked. The antigen is sandwiched between. In this picture, we have an antibody bound to the petri dish. We have a detection antibody that will sandwich the antigen. And then we finally have our secondary antibody that contains the horseradish peroxidase that will create our color. So that's a summary of the three that we discussed. In lab, you're going to use this 96 well plate, and you're going to be looking for a specific color. If the color is present, that means that one of those reactions took place. Either the antibody was bound to the plate, or the antibody was bound to the plate. And in the end, some type of antibody that was linked to an enzyme bound to the antigen, and a color was created. Next, we will look at blood typing. Blood typing is sort of related because antibodies can be produced. As you may know from general biology, there is 4 types of blood based on the ABO blood grouping. But we only have 3 alleles. 2 alleles are codominant to one another, and 1 is recessive. The first one is big I superscript A. If your red blood cell has that allele, you will produce a sugar on the surface of your red blood cells. We're going to label that A. If you have an allele big I superscript B, you will produce a sugar that's on the surface. We're going to label it B. If you have the little I, and you must have 2, you'll produce no sugars. Because of the variations that can be produced from these 3 different types of alleles, it gives rise to 4 different blood types. The first patient has blood type A, so they have the sugars A. The next person has blood type B. Blood type AB would have both sugars, A sugar and B sugar, whereas blood group O will not have any sugars. This blood group is the universal donor. Since it doesn't have any sugars, then your immune system will not see it as foreign, and your immune system will not develop antibodies to attack it. In this slide, we're going to talk a little bit more about the antibodies. So let's look at blood group A. Blood group A, that patient will produce antibodies against B. So they produce antibodies B. Now, a person that has blood group B, they will produce antibodies against A. So you can't give that patient blood group A because they're going to produce antibodies against A, and their body will immediately destroy those blood cells. A person that has blood group AB, they're not going to produce any antibodies. Therefore, they can receive all type of blood groups. They can get A because they don't produce antibody B. They can get blood group B because they don't produce antibody A. And then finally, we'll look at blood group O. This patient produced antibodies against A and antibodies against B. So this patient can only receive blood group O. Based on the ABO system, only O group can be the universal donor, but they cannot receive anything but O. So let's dive into that to make sure you fully understand. We're talking about a patient that has O blood. They have antibodies for A and they have antibodies for B. So if you give them a patient that has A blood, they donate some blood to that patient that has O. What's going to happen is you're going to have an interaction of an antibody to an antigen. You gave the patient A. The person with O has antibodies against A. The antibody is going to clump together with the sugar molecules of the blood cell and form these clumps that we call opsonins. Let's say you give a patient with O blood type B blood. The same thing will happen. They're going to produce opsonins. And if you give a patient with O, AB, they are also going to produce opsonins. So a person with O can only accept O blood. O blood, just like the other three types, can be further separated into Rh positive and Rh negative. This is the Rhesus factor, another molecule that's located on the surface of the blood cell. Based on the Rhesus factor, only group Rh negative can be a universal donor. Notice that the Rh negative doesn't have any molecule on the surface of the red blood cell. Therefore, technically, the universal donor has blood group O and they are also Rh negative. That is the only one that is the universal donor. This slide shows other molecules that are also on the red blood cell. Here is the membrane of the RBC. This is the red blood cell's membrane, and you have this HH factor, you have the MSN, MNS factor, you have the Buffy factor, you have the RH factor, you have many different types of proteins and sugars that are bound to the membrane that creates blood grouping. The only one you need to worry about is the ABO blood cyst grouping and the RH blood grouping system. Approximately 45% of Caucasians are type O. Some are positive and some are negative. But 51% of African Americans and 57% of Hispanics are type O. Minorities and diverse populations also can contribute to the blood bank. Therefore, they play a critical role in meeting the constant need for blood. Type O negative and O positive are in high demand. Only 7% of the population are O negative, which is the universal donor. However, the need for O negative blood is the highest because it is used most often during emergencies. The need for O positive is also high because most of the population has that blood group. They make up about 37% of the population, so they would need O positive blood.